Hi and welcome to everyone to join here the Myco-Talks today. My name is Oliver Kurzai and I'm very happy to welcome you together with my co-chair Jay Kolls, who you can also hopefully see in the camera. We are both excited to chair this Myco-Talk session today, and before we start with the two speakers, let me give you a few words of introduction. Some of you might have been to other Myco-Talks before, others may be new in the audience, and we just want to point out that this is a lecture series that covers medical mycology, all aspects of medical mycology, with two 30-minute talks in each session. Of course, all speakers have been selected because of their excellent work in science or medicine having to do with fungal infections, but we also put a focus on diversity. And specifically today, you will see that we will have a very good gender aspect. So Jay and me stay in the back and two women give the talks, also career stage, geography and field. Now, some things that you should know: there is a Q&A function, and we invite you to submit questions also already during the talks. And these questions will be seen by Jay and myself, and we will then sort this and ask the questions in the end to both of the speakers. We hope for a lively discussion, so please feel free to submit any questions or comments that you would have. And for everyone who wants to see this again, all the talks are actually available at the MRC CMM YouTube channel, and you'll find the address here. Now, we have two speakers today, and we will introduce them in detail right before their talks. We will start with a talk from immunology given by Sarah Gaffen, and then we will have a talk on Candida albicans, cell and molecular biology, given by Alex Brand from Exeter. And I also want to mention the organizing committee. So, of course, this is an activity done by the Center for Medical Mycology, the Medical Research Center at the University of Exeter. But there is an international organizing committee, and you can see all the members below. And they have given some input with regard to who should be speakers on this list. But most of the work is actually done by the colleagues in Exeter, and we are very grateful for that. Of course, this is not going to be the last session in this lecture series. It's actually growing and growing, and I just learned that this is again a record number of attendees at this time for the lecture series. And we continue with highly renowned speakers on the 19th of November with Joe Heitman and Tobias Hall. And then in December, shortly before Christmas, Judy Berman and Michael Leonakis from the NIH will give a talk. So make sure that you register for each of these sessions, and this has to be done independently for each session. So with this, I'll stop the introductory slides and hand right over to Jay, who's going to introduce the first speaker, Sarah Gaffen. Jay.
Great. Thank you so much, Oliver. So it's my pleasure to introduce my friend, collaborator, and colleague, Sarah Gaffen from the University of Pittsburgh. Sarah did her bachelor's degree at Carnegie Mellon, and then PhD at the University of California in Berkeley, followed by a postdoc at the Gladstone Institute, where she studied cytokine receptor signaling. And then her first faculty position was at the University of Buffalo, where she started work on a novel cytokine receptor at the time, IL-17 receptor, and she's made fundamental insights into IL-17 receptor signaling as in addition to other IL-17 receptor family members. And it was also during this time that she first made the discovery that IL-17 receptor was critical for oropharyngeal Candida susceptibility. She moved to Pittsburgh and has continued to make seminal insights into both the basics of receptor signaling in the IL-17 receptor family, as well as insights into how IL-17 receptor and other cytokine receptors mediate host defense against oropharyngeal candidiasis. So today's talk is titled "A Tale of Two Cytokines: How IL-17 and IL-22 mediate host resistance against Candida albicans in the oropharynx." So welcome, Sarah.
Thank you, Jay. Get my screen going here. Okay. Thank you so much. It's really great to be here. Welcome to everybody. Alex and I were both saying before this, it's actually a little nerve-wracking to do this to an invisible audience. So without further ado, let me get going. This is my lab, who of course are really the people who do all the work. And when I say we, I'm referring to them and our various collaborators. I do want to make the point before I move on that everybody in my lab and myself are all either in the US, either immigrants or descendants thereof. And as we sit here at the end of October on the eve of the most significant election, presidential election in US history, and I say that with no exaggeration, to any Americans in the audience who are eligible to vote, if you have not done so, please do. Keep in mind that we are citizens of the world in addition to being scientists, and we need to sometimes get ourselves out of the lab and doing things that matter to us. So with that, let me move on. I do want to thank Jay, especially for that introduction, because he really set up the background to which I got started in this. I was not trained as a fungal immunologist. I really didn't know very much about fungi until I did a postdoc at the Gladstone Institute at UCSF. And became much more aware of the importance of opportunistic fungal infections in the context of HIV/AIDS. I've just got a picture here of the very first report describing AIDS. And of course, these patients had pneumocystis pneumonia, which was the basis of the report. But I've underlined the fact that they also all had Candida infection. And oral candidiasis is one of the most common AIDS-associated infections. It is, in fact, an AIDS-defining illness. Although, of course, it's not potentially the most serious disease that people have, it really begins to illuminate, I think, many principles of mucosal immunology, especially in the oral cavity. And it's also worth pointing out that although Candida causes diseases at many surfaces, by far the most common in HIV patients is oral candidiasis. And by implication, that suggests that CD4 T cells have an especially important role in the oral cavity. And that's really sort of how I ended up getting into looking at oral candidiasis and IL-17. Now, when I was doing my postdoc and early in my career, we only knew about two kinds of T helper cells, CD4 cells, Th1 and Th2. And that paradigm underwent a major change in 2005 with the discovery that IL-17 was the characteristic cytokine of a new subset of CD4 T cells. I had been working on IL-17 since 1999, and so as had Jay, and that's how we know each other. And it was already clear that it was a pretty fascinating cytokine. And so sort of putting all those things together, it became logical for us to begin to look at some of the AIDS-associated diseases like oral candidiasis. So what is IL-17? I was drawn to start to look at this because the cytokines and the receptors are structurally and biochemically distinct from other known cytokines. So they're not interferons, they're not TNF type receptors, they're not TLRs or IL-1 receptors. And so that I thought would be fertile ground for beginning to understand a unique type of cytokine family. So when I say IL-17, I really mean IL-17A for the most part. This is the founding member of the family. But type 17 cells also make IL-17F, which can form homodimers, but also heterodimers. So type 17 cells, the most famous, of course, are Th17 cells, which are CD4 positive T helper cells, but many other lymphocytes also make IL-17, both in the adaptive immune response, but also in innate immunity. And I'll touch on some of this as we go forward. IL-17, A, F, and AF all use a common receptor that is expressed predominantly on non-hematopoietic cells. And these are really the major responders. And I'll talk about that principle in this talk. And type 17 cells also make other cytokines like IL-22. And its receptor, again, is expressed predominantly, although not exclusively, on non-hematopoietic cells. And together, these cytokines mediate what we consider to be a type 17 response. IL-17, I think, came onto a lot of people's radar because of its role in driving autoimmunity. And that prompted development of biologics that target IL-17 or its receptor. And these were approved a few years ago and have been quite successful, especially in psoriasis. And part of my lab focuses quite a bit on that aspect. But of course, IL-17 did not evolve to give us autoimmunity, but rather plays a key role in host defense and tissue repair. And I will be telling you a little bit about the story of how we showed that IL-17 receptor signaling was important in oral candidiasis. Okay, before I do any of that, I want to tell you a brief story. This is about Rachel Bailey, who was an undergraduate in my lab and later did a year as a technician. And actually, it's really a story about her mother. And Rachel's mom had lifelong psoriasis, psoriatic arthritis, very severe, poorly controlled. And most drugs did not work terribly well for her until the advent of the anti-IL-17 biologics. And so things got much better for her at that point. Her plaques cleared, she felt better. And I wish I could end the story there, but unfortunately, she experienced not only recurrent oral candidiasis, but also an inflammatory bowel disease, which is becoming, unfortunately, a common side effect of these biologics. And I think this ties together in a single individual these various principles of what IL-17 does and can do in the context of the body. So as I said, we got into this from the point of view of being interested in T cells and their roles in oral immunity. And when I say we, I really mean Heather Conti, who was a graduate student in my lab at the time. And what Heather showed was that mice lacking either subunit of the IL-17 receptor, we started IL-17RA, its partner IL-17RC, renders mice susceptible to oral candidiasis. And this was a wonderful collaboration with Mira Edgerton at Buffalo and Scott Filler at UCLA. And you can see that mice develop plaques, they lose weight, whereas wild type immunocompetent mice are fine. And that was great for us. We had a system, we could begin to study this. Oh, I will also mention the adapter ACT1 that is used by the IL-17 receptor also is required for this response. But very gratifyingly, Jean-Laurent Casanova and Anne Puel had identified patients with chronic candidiasis. And it turns out that a number of those cohorts had mutations in the same molecule. So the IL-17 receptor A, IL-17 receptor C, or ACT1. Candidiasis is actually one of the dominant diseases that these patients get. They do get some cutaneous staph infections and others, but candidiasis is a very dominant effect in these individuals. And that really told us that we were on the right track. I'm going to tell you a series of vignettes today about understanding IL-17 and also some of the other type 17 cytokines that are involved in candidiasis. And I'll start with where IL-17 acts. I intimated a bit earlier that a lot of our interest is in the downstream IL-17 signaling pathway. And so it's important to understand what the right cells are to respond to this cytokine. It was already well known from work from Jay and Eric Leberts and many others that IL-17 seems to act dominantly on non-hematopoietic cells. And in the oral mucosa, this has a very interesting epithelial biology. It's a stratified epithelial layer composed of a superficial and intermediate layer that gets sloughed as part of an ongoing infectious response, and an underlying basal layer that is proliferative and can undergo a differentiation process that then replenishes this superficial layer. And these two layers are characterized by expression of distinct keratins. The basal layer is characterized by expression, for example, of keratin 14, which many in the audience may be familiar with. It's the CRE for this gene is often used as a way to delete genes in epithelial surfaces. The superficial layer is characterized by expression of keratin 13 as well as keratin 4. And these have a much more restricted expression, including the oral, esophageal, and vaginal epithelia, which coincidentally or not, are tissues that IL-17, Candida likes to infect. Now, you can see in this image that the IL-17 receptor is expressed almost exclusively in the superficial layer, and that prompted us to create CRE-expressing mice driven by the keratin 13 promoter. And these CRE mice work really well. You can see a LAC-Z reporter is expressed very specifically in that superficial tissue. This is the surface of the tongue. We then crossed those mice to an IL-17 receptor flox allele, kindly given to us by Jay, and assessed the susceptibility to candidiasis. And I think what you can see is that whereas wild-type mice clear Candida very rapidly, IL-17 receptor total knockout mice have a high susceptibility, as I've shown you, and mice lacking the IL-17 receptor just in that superficial layer show a pretty similar phenotype to a complete body knockout. And this is true for fungal load as well as for weight. If we look at the genes that are affected in these knockout mice, they're very similar between a total knockout and a keratin 13 specific knockout. In yellow, these are a very small subset of the genes that get induced in a wild type mouse upon infection very early in the game, and various genes that are lost in the absence of the IL-17 receptor. These are pretty characteristic of the typical IL-17 gene signature, work done by us and Chen Dong and many others, and they give us some insights into what IL-17 is doing in the context of Candida. So for example, one of the genes that we always see when we look in these settings is a very high induction of beta defensin 3 that is almost entirely lost in these knockouts. And indeed, mice lacking beta defensin 3 are very susceptible to Candida, almost as much as a total 17 receptor knockout. We also see genes associated with the neutrophil response. So chemokines like CXCL1, CXCL5, or GCSF. And indeed, in our hands, IL-17 receptor deficient mice show a very severe diminution in recruitment of neutrophils to the oral cavity after infection. IL-22 knockout mice don't, and I'll come back to that a little bit later. But for the students and trainees in the audience, I want to point out, not every gene on this list actually does participate in the response. So just because a gene's induced doesn't necessarily mean that it's a driving effect. So for example, CCL20, lipocalin-2 are also two genes we always see, but yet we've shown that those don't seem to be important, at least in this early response. So where is the IL-17 coming from? I'm just going to summarize quite a lot of work that we and others in the field have done on this, because I want to move on to more recently published work. But what Heather Conti and Akash Verma in my lab showed using reporter mice and a variety of other studies was that IL-17 comes in this model system dominantly from innate type T cells. And the reason that they're innate is that mice, unlike humans, are naive to Candida albicans. And so when we are infecting them, this is actually their very first encounter with this organism. But nonetheless, IL-17 is turned on within 24 hours. And I already showed you that the clearance happens within 2 to 3 days. And so clearly this is an innate response. Now, interestingly, IL-17 comes both from gamma delta cells, which was not surprising. These are well-known innate cells at mucosal surfaces, but even more prominently from a CD4 positive population of T cells that express what would normally be considered a conventional T cell receptor. And these are sometimes called natural Th17 cells. And not only do these cells express express IL-17, but it's these CD4 T cells that proliferate upon exposure to Candida, whereas the gamma delta cells, although they are there and making IL-17, do not proliferate at all. And this contrasts with what is seen in the skin, where gamma delta T cells are the dominant source of IL-17 in dermal candidiasis and do undergo a proliferative response. And so if we interrogate the activation of these cells a little bit, what we found was consistent with the fact that these seem to be operating in an innate manner, that there's no requirement for what we would consider to be typical adaptive Th17 signals, like CCR6 or CCL20, for example, or IL-6. There's no ongoing T cell receptor signaling. Any TCR will do, and IL-1 is required. And I'm not going to dwell on this. I'll just say that a lot of work from various people has contributed to this, and we can talk about it in the Q&A if people have more questions. Okay, so how are these T cells activated from the point of view of Candida? Well, I don't need to introduce the fact to this audience that Candida is a dimorphic fungus, and it's the hyphal form of Candida that is virulent. And if we infect mice with a yeast lock form of Candida albicans, we see no activation of IL-17 in the oral cavity, whereas the reconstituted strain activates it just fine. So we thought, great, okay, Gordon Brown and many others in the Dectin field have shown beautifully that Candida hyphae are sensed by C-type lectin receptors. And so that's the answer. Now, just a brief reminder, hyphae, particularly beta-glucans, are sensed by dectins and other related C-type lectin receptors. They signal through an adapter called card9, and TLRs, especially TLR2, have been also implicated in this pathway. So you can imagine our surprise when we infected mice lacking these various components, and they all cleared Candida just fine in this innate setting. Sorry, this early acute setting. Again, whereas 17 receptor knockout mice don't. IL-17 expression was perfectly normal in those mice. IL-22 is expressed normally. The proliferation of the natural Th17 cells was normal. And so this pathway did not seem to be operative in that early response to Candida. So we're really left scratching our heads, but I ended up doing a sabbatical in London in 2015 to 16 with Julian Naglick, and that shed some light on how this is working. And this was work done in Julian's lab, but in collaboration with Bernie Huba, who gave a beautiful Myco-Talks, I think last month, and with David Moyes and John Richardson in Julian's lab. And so what they had discovered was that when Candida becomes hyphal, it secretes a pore-forming peptide, which they called Candidalysin. This is made in the invasion pocket, and it induces damage in the oral epithelial cells. I'll just emphasize that Candidalysin, encoded by the ECE1 gene, is expressed only in hyphae, but it's not required for hyphal formation. So beta-glucans are still exposed, even in Candida that lacks Candidalysin. The action of Candidalysin triggers cell damage through inducing a CFOS signaling response. And CFOS also leads to upregulation of various genes that are associated with an epithelial innate immune pathway. Now, these are genes like IL-6, IL-1, CCL20, beta defensins, all of which are genes that, from the lens of somebody who works on IL-17, are really connected to type 17 immunity. And based on that, we thought, well, wouldn't it be interesting if the very initial response to Candida is through Candidalysin rather than through the Dectin pathway? And after that build up, it would be sad if I told you it wasn't. So indeed, Candidalysin is required for proliferation of these TCR beta cells, as shown here. Candida lacking EC1, the Candidalysin gene, have a significantly impaired response to induce IL-17. Moreover, beta defensin 3 is also impaired in the absence of Candidalysin, as is the activation of the neutrophil response. And this is all measured very early, days 1 and 2 after acute infection. We saw no change in gamma delta cell frequency. Fungal loads were very similar in this early time point, so it wasn't simply a question of fungal load. And I should point out, not all virulence factors do this. For example, the SAP46 complex activates IL-17 just fine. Not only that, but IL-17 and Candidalysin both act directly on the oral epithelial tissue. So I've just told you Candidalysin acts on this tissue to induce CFOS. IL-17 acts on these cells to induce an NF-kappa B response. And these have a synergistic output on the target genes of interest. So you can see, for example, if we use CCL20 as an example, IL-17 and Candidalysin both induce it, but together they induce this cooperatively. And for all of these genes, we saw either an additive or a synergistic induction with these two signals together. So everything I've told you so far really focused on IL-17A, but what about some of the other members of the family, particularly IL-17F? This is work done by Natasha Whibley, who is now a postdoc at Oxford. And again, just as a reminder, type 17 cells make F, which is a homodimer, but also forms heterodimers with IL-17A. Now, there's a lot of data on this slide, but let me walk you through it. These are just controls. IL-17F knockout mice, or mice in which we neutralize IL-17F, are not susceptible to Candida. However, neutralization of IL-17A does render mice susceptible to candidiasis, and the same is true for a knockout, which I'm not showing you. Neither of these are as potent as a complete IL-17 receptor knockout, or ACT1, which is equivalent, as you can see here. However, if we neutralize IL-17A in if we neutralize IL-17F in IL-17A knockouts, this renders mice far more susceptible than blockade of either alone. So these two cytokines, despite using the same receptor, are acting in a functionally synergistic manner. Further insight that this might be relevant in people came from the work by Casanova and Puel, in which not only did they identify patients lacking the 17 receptor, but they also identified a kindred in which the IL-17F gene was mutated. It's a serine to leucine mutation at position 65, and many of the affected individuals have candidiasis. Not all of them, I will say, but most of them do. And beautiful in vitro studies in this paper showed that the IL-17F mutant, the S65L mutant, had dominant negative activity, not only to IL-17F, but also to the IL-17A/F heterodimer. So this mutation is indicated here, and it interferes with the binding of IL-17F to its receptor. And this sequence is very highly conserved in the mouse. And so based on that, we created mice that have the same mutation. Actually, first we made an IL-17F mutant in vitro and showed that it had reduced signaling capacity. So you can see wild-type IL-17F induces signaling. It's very weak, by the way, even compared to IL-17A, which itself is not super strong, but we do see some signaling. And this mutant is largely abrogated in signaling, except at very high doses, which is commensurate with how we think that this works. So we used CRISPR-Cas9 to make knock-in mice, and it worked great. In fact, half of our founders were homozygotes, and subjected them to candidiasis. And again, there's a lot here, but what you can see is that wild-type mice clear Candida. Mice that are heterozygous for this mutation, which would be the most similar to humans, also clear Candida. So it doesn't seem to be having dominant negative activity in vitro. However, mice that are homozygous for this mutation are susceptible at a level equivalent to an IL-17A knockout. And the N is very high here because we really wanted to be sure of what we were seeing. Again, this is less than what we see with an IL-17 receptor or an ACT1 knockout, but nonetheless, these are very susceptible. Now, the susceptibility that we saw was associated with reduced chemokine expression, like CXCL1, and consequently reduced neutrophil recruitment to the tissue. Not completely abrogated, but certainly impaired. But interestingly, there was no effect that we could see on beta defensin 3, which we know is an important component as well, and that may be why it's not as significant an effect as we see in the total 17 receptor knockout. And this was work done by Chin-Cheng Zhu and Laetitia Monin. So in the last part, I want to talk about another cytokine, IL-22. Now, Felix Agor was a graduate student in the lab, now a postdoc, who took this on. And I sometimes joke that he went where few in the Gaffen lab dare to go, which is to ask if IL-17 might not be the only thing we should be looking at. So you can see IL-22 knockout mice are very susceptible to candidiasis, almost as much as the 17 receptor, not quite, but pretty close. So this cytokine itself is important. And let me remind you that IL-17 and 22, though made by the same cell types typically, are very different. 17 receptor, as I said, is a unique family, quite structurally distinct from other cytokines like the IL-22 receptor. It uses ACT1, it activates NF-kappa B and other pro-inflammatory pathways, leading to expression of acute inflammatory genes like CXC chemokines. IL-22 is a JAK-STAT activating cytokine, and its main stat is STAT3, and the genes that it tends to be associated with in many other tissues are genes involved in wound repair, proliferation, tissue healing, that sort of thing. So Felix first asked, are these cytokines redundant in candidiasis? And we did this in collaboration with Mihalis Leonakis. And you can see that this is actually unpublished data. If we cross a 22 receptor knockout to an IL-17 receptor knockout, the mice have a higher fungal load, about approximately a log, compared to either any knockout alone. And we saw exactly the same thing if we neutralize IL-22 in a 17 receptor deficient setting or neutralize 17 in a 22 deficient setting. So where is IL-22 acting? Well, let me just refresh your memory. And we already showed you that IL-17 receptor acts in this superficial keratin 13 expressing tissue. And so we dually crossed IL-22 receptor flux mice, again from Jay, and there was absolutely no phenotype. So 22 receptor in this tissue doesn't seem to matter at all. However, it turns out it's actually even more highly expressed in the basal K14 expressing layer. And indeed, when Felix crossed the 22 receptor flux mice to a K14 Cree, these mice were susceptible. And we saw exactly the same pattern for a STAT3. So it had no effect in the keratin 13 expressing layer, but was critical in the K14 layer. So what is IL-22 doing? As I said, that IL-22 drives proliferation. And so using a Ki67 stain, Felix looked at the keratin 14 layer, and the K13 layer as well, and showed that in the context of oral infection, there's a very high proliferation of this K14 layer. And this is almost completely absent in a 22 receptor knockout. And I was surprised by this because I would have expected that even if IL-22 contributes to proliferation, it wouldn't be the only thing that's important. And yet, as far as we can tell, it seems to be completely, almost entirely non-redundant for this effect. What was even more surprising was when we looked at the genes that were IL-22 dependent, so this is RNA-seq comparing wild type to a 22 knockout, a lot of the genes that we saw were those associated with an IL-17 gene signature. And this is looking at total ton. And among those genes was the IL-17 receptor itself. And you can see by staining, here's the IL-17 receptor in the superficial layer. It's there in a wild type after infection, but it's absent in a 22 knockout. And yet, 22 and 17 seem to be acting in different tissues. So what does this mean? Well, the model that we think best fits all of this data is that IL-22 acts on the keratin 14 basal layer and through STAT3 drives a proliferative response that then replenishes the superficial layer. This is where IL-17 receptor is. This is what drives the IL-17 receptor dependent genes. And therefore, even though acting on spatially distinct areas, these cytokines functionally interact. Or in other words, IL-22 licenses the oral tissue to be able to respond to IL-17 receptor. Or if we put this another way, this is my second to last slide. When Candida becomes hyphal, goes from yeast to hyphae, it secretes Candidalysin. This activates a damage response through CFOS that leads to induction of various cytokines. Some of these factors have direct anti-fungal properties. They also activate neutrophils that further help clear the fungus. Some of these cytokines activate innate type 17 expressing cells. I didn't really talk about the roles of IL-1 or IL-36, but I can in the Q&A. Some of these we think then activate the adaptive response as well. And regardless of the source, IL-17A, F, and 22 act on distinct tissues in the oral mucosa, with 22 activating STAT3, driving proliferation and replenishment of the superficial tissue, IL-17 activating NF-kappa B. And as I showed you, because these seem to act cooperatively, Candidalysin and IL-17 further stimulate expression of these genes. And this whole process is only put into play when Candidalysin is present, meaning that the tissue is damaged. So the tissue can distinguish not just between hyphae and yeast, but between damaging hyphae and benign hyphae. And this is probably a survival advantage for commensalism, which we know is important for maintaining immunity, not just to Candida, but probably to other fungal species as well. And with that, I'll just thank all the people who did the work. I think I acknowledged them as I went along, so I won't spend too much time with that. I do want to give a shout out to the Myco-Talk crew in Exeter who have made this wonderfully smooth and seamless, and my funding sources. So with that, I will stop.
Thank you, Sarah. So Oliver, we'll do questions after the both talks.
Yes.
Perfect.
Right, so I'm supposed to go on, but the host has blocked my video. Thank you very much. Okay, so as Jay said, we will do the Q&A afterwards. Already some questions popped in, so please keep on sending in your questions. We'll try to sort this for Sarah in the end. And now it's my pleasure to introduce the second speaker of today, and this is Professor Alex Brandt from Exeter. And she is a biochemist and microbiologist by training, and her work focuses on Candida albicans and some very specific aspects of Candida albicans, molecular and cellular biology, I would call it. But always when I listen to the talks that she gives, it's about questions that other people seem to ignore. And this is questions like, how does Candida grow and in which direction does it grow and how is this possible? And also her talks are always connected to wonderful videos. So I'm very much looking forward to seeing this, Alex. Yeah, that's about it for the introduction. So I invite you to share your screen and then we're looking forward to your talk. Thank you.
Thank you very much, Oliver. Okay, thank you, everybody. I'll just start my laser pointer. Okay, so I work on Candida albicans, and my talk today is going to be about RSR1, which is a small GTPase. And RSR1 has been considered as a polarity factor in Saccharomyces cerevisiae, where it's often referred to as BUD1. But we've found that actually it's like its counterpart in mammalian cells. RSR1 is more like RAP1, and it's a major organizer of the spatiotemporal processes that go on within cells. So I'm going to show you this video. This is made by Darren Thompson. And in this, I want to point out the way Candida albicans hyphae grow. And after this video, I'm going to talk about a little bit of phenotype that we've done before with Darren, and then the rest of the work in this talk is going to be that done by Tina Bedekovic, who's a postdoc in my group. So to play this video, I want you to take two things from this. Firstly, I want you to notice that growing hyphae are very dynamic, and the nuclei that you can see with GFP tagging there are move around a lot. And you can also see the septins in GFP as well. And what actually happens here is that even though with all this movement, albicans forms a string of mononucleate cells, and that takes very careful organization of the processes inside the cell. So why are we so interested in hyphae? Well, as Sarah was admirably describing, hyphae are the invasive form. And although we see yeast in mucosal biofilms, vaginal biofilms, it's the hyphae that are underneath these cell layers that really do all the damage and set off the inflammation response. But hyphae are also responsible for biofilms on medical devices, and they can even grow down into the substrate, which is the polymer that the device is made of. And this renders them A, dangerous if it's in a catheter setting, but B, if something like this voice prosthesis, the hyphae make the device swell up and cease to function, so it needs to be changed. But of course, we are mainly concerned with the deaths caused by Candida albicans, and you can see the lesions here in this mouse kidney. And within the lesions, of course, there's a lot of hyphal growth, and it's the presence of invasive hyphae that trigger the hyperimmune response and lead to organ failure and death. And of course, hyphae are the escape mechanism from our immune cells. And as I was pointing out with the medical devices, albicans can grow in any part of the body, and its hyphae can even invade cartilage in joints. So it's a determined beast. So in our previous work that we were looking at hyphal growth and the role of RSR1 in this, we tagged the Spitzenkörper MLC1, and you can see that as this little fluorescent dot here. And what we found when we were looking at wild-type cell growth was that albicans hyphae like to grow under contact surfaces. And when they do, they sense contact, and their growth machinery then skews towards the point of contact. And this then leads to a number of growth behaviors that one could argue are important for tissue invasion. So the first one that we see here is contour following. So hyphae seem to be able to sense that they're on a surface, and they follow that contour rather than meandering off away from it. And this becomes very important for gap penetration. And if you watch this movie, you'll see the hyphal tip trying to penetrate the substrate underneath it, the surface that it's on, and that allows it to find a gap to grow down into. And then lastly, trajectory maintenance, which seems to be very important for continuing down into the substrate beneath the hypha. So you'll see that the hypha sets off and it can grow round an obstacle and then carry on in the trajectory that it first started in. So another term for this is directional memory. So we started off with Cheryl Gale deleting RSR1 and looking at these responses in the RSR1 mutant. What we find is that the Spitzenkörper is central within the tip, and the cells don't seem to sense the surface that they're growing on. and they can't follow contours as a result. And you can see here this aberrant hyphal growth. And when they come to this corner or gap here, they just don't see it. And so they seem to be blind and not able to respond to the physical features of their environment. And this leads to an old paper by Yar et al. In 1977, where they found that RSR1, the mutant, is actually avirulent. And when looked carefully at the tissue burdens of with this mutant, what was happening was that the cells were circulating around in the bloodstream, but they couldn't escape from the bloodstream into the deep-seated tissue below. And with Cheryl Gale, again, we did this experiment where we grew wild-type cells on top of a cell layer, and you can see that in the wild-type Candida cells, they disappear down into the layer below, whereas the RSR1 mutant can't penetrate the substrate and it just grows on top, and so you retain this fluorescent signal. And as a result, the RSR1 mutant doesn't cause tissue damage. So how are we going to investigate the regulation of this GTPase further? So just to do the naming of parts, which we always have to have when we do molecular work, RSR1, this GTPase, is controlled by its activator, the Bud5-Gef, and its deactivator, Bud2. And RSR1 in the model with Saccharomyces, and certainly in albicans yeast, localizes CDC24, which is the activator for CDC42. So CDC42 is highly conserved across all eukaryotic systems as a polarity protein, but it also does a number of other things. And soon we'll be hearing more about its activity through the CLAR4 kinase. And this system here has a role in vacuole fusion and in septin ring assembly. So RSR1 GTPase sites the bud site next to the previous bud scar in yeast, but in hyphae it doesn't do that. In fact, hyphae are free to germinate in any direction that they care to, and they use this to respond to external signals. So the next thing I need to do to you is explain how normal hyphae grow in albicans, so that then you can see the difference in the RSR1 mutant. So our late colleague, Nick Reed, described albicans as a filamentous yeast, and I think that's a very good description of it. And it grows by this method called apical dominance. And this was defined for albicans by Gao and Goudet in 1982. But it's a common feature of tip growing systems. And so it's the same that it happens in plants. So we'll get to that in a second. So albicans starts off as a germinating yeast cell with a fairly small vacuole, one nucleus, and the Spitzenkörper in the tip. Then what happens is the vacuole starts to expand and pushes the cytoplasm forward into the germinating hypha. And then the start signal occurs, which sets off chromosome duplication and sets down the nascent septin rings just behind the tip. Then the vacuole increases further, pushing more cytoplasm into the tip as it grows, and new vacuole vesicles bud off, and they start to be pushed into the new hyphal tube. And here we have the final step in nuclear division. We've now got two nuclei. So again, the vacuole is expanding, more cytoplasm into the hypha, and now the daughter nucleus has passed into the through the presumptum, or that's the open septum ring, as well as to the little vacuolar fragments. Then later on, the septum will form pretty much as soon as this nucleus goes through the septum closes, and the vacuoles start to fuse and enlarge. And in the end, you get a string of cells where you've got cells at different stages. So the apical cell is growing as fast as it can. It's full of cytoplasm, and soon these vacuole fragments will start to fuse. Here in this quiescent cell, we've got vacuoles that have fused and expanded, and they're taking up most of the cell compartment bounded by the two septa. And here in the original mother cell, the vacuole started to shrink because new nutrients have been taken up and a new hypha is able to emerge. So you can see in one whole view of your microscope, you can see cells at all different stages of the cell cycle. So what's the role of RSR1 in this? Well, RSR1 in albicans is N-RAS-like, so unlike in RSR1 in Saccharomyces. So that means it has these two cysteine residues at the C-terminal, and you can see them here marked in the membrane interacting domain. And one is palmitoylated, this first cysteine, and the second one is farnesylated. So if we look at the process of RSR1 cycling in cells, after translation, RSR1 with its palmitoylation and farnesylation sites at the C-terminal are translated in the cytoplasm, and the farnesylation signal acts straight away in the cytoplasm so that the the acid tail is added. This directs RSR1 to the ER, the secretory pathway, and this gets to the Golgi. At the Golgi, this second cysteine is palmitoylated, and that stabilizes RSR1 in the vesicles that then lead to the hyphal tip and the polarity site. And there, it's activated by BUD5, the GEF. And its activity the range of activity is regulated by these two gaps, which form a collar around the hyphal tip to limit the site of activity in the hyphal tip. Now, as long as RSR1 stays palmitoylated, it will stay in the plasma membrane. But it can be depalmitoylated, and then it will be transported probably by VPS35, because we pull that down with RSR1, and it will then go to the endomembranes. And the cycling within this intracellular domain is a sort of up for discussion at the moment in all eukaryotic cells, but we think it probably rotates around this system. So we would expect to see a YFP RSR1 at the whole of the cytoplasm, at the whole of the membrane, and that's indeed what we do see. So if you watch this movie here, you can see the vesicles bearing YFP RSR1 are highly dynamic in hyphae. And if we look at them in these images here, we can see that RSR1 is throughout the cytoplasm, possibly with a bias towards the hyphal tip. And we can see, as per the model, BUD5 is in the tip, and BUD2 forms a collar to deactivate RSR1 as it moves away from the area where it is required. So we made a bunch of mutants, and we're going to be looking at those in a while, but we'll just look at their localization to start with. So we made the palmitoylation mutant, and as we suspected, the farnesyl moiety is still able to take RSR1 to membranes, but it's not stabilized at the plasma membrane, and instead it ends up in the endomembranes, particularly the vacuoles. But if you delete the farnesylation site, you end up with RSR1 entirely in the cytoplasm because it never gets into the secretory pathway. When we made mutants where we have RSR1 permanently GDP bound or GTP bound, we find that neither of these affect the ability of RSR1 to get to the membrane. But what we do find is that the RSR1 GDP bound version can actually account for the bias of what we saw for the wild type in the tip. So RSR1 GDP does tend to be in the apical cell of the hypha. So now I'm going to take you through some of the mutant phenotypes that we looked at in the RSR1 null. And then I'm going to show you how, by expressing the mutants that we made in the RSR1 background, we were able to rescue some, but not all, of the phenotypes. And using this mechanism, we've been able to dissect how the regulation of RSR1 feeds into distinct processes within the cell. So as expected, if you delete RSR1, you get bud sites all over the place in yeast. And in hyphae, the hyphae don't invade the substrate, as we've already seen. And here we've got the reintegrant, and here's the reintegrant, and here's the YFP RSR1 rescuing the phenotype, wild type phenotype. So then we looked at septins, because CTC42 and CLAR4 are known to organize septin ring formation. So in wild type, you can see the septin rings separate into 2 rings, and then the septum can form between them, and we can see this with calcofluor white staining. But in the RSR1 mutant, we find that we get a whole host of aberrant septin ring formations. So here, instead of 2 septin rings separating, we have 3. And then this results in 2 distinct septa. You can see those here in calcofluor white. Or we get an aggregation of septins at the side of the cell. And again, here we can see it in TEMs and with calcofluor white staining. And we even see septa trying to form in the mother cell. And here are the septins. And gradually you can see the blue of the chitin coming in to try to form a primary septum. And then this appears as a blister on the side of the mother cell in transmission electron microscopy. So if we've got septin rings not forming properly, what does that mean in terms of the cell's ability to close its septa, which is very important for our mononucleate compartments? Well, we looked at the DBF2 kinase because that, as you might be able to see here, it travels on the spindle pole bodies during nuclear division, and then it translocates to the septa from where it directs actomyosin ring contraction and also building the primary septum with CHS2. And you can see it here at the septum in wild-type cells. But in the RSR1 mutant, we see that although it is recruited at septa, it forms this cloud around the septin rings, but septa do manage to finally close. And we were able to determine that what happens here is that the cloud, although it does work, it delays the whole septal closure process. So this is septal closure in wild type cells, and this is much slower in the RSR1 mutant, and you can see the time difference here. So then another function of CDC42 and CLAR4 is in vacuole fusion. So we looked at vacuoles, and here they are in the control cells, and you can see the vacuoles take up nearly the whole hyphal compartment in the mutant, in the reintegrant and in the control. But in the RSR1 mutant, the vacuoles do not fuse. Instead, they stay as fragments. And if you analyze these images and look at the amount of cytoplasm that are retained in these subapical cells, you'll see that it's greatly increased the amount of cytoplasm that remains. So we wondered whether this would mean that these cells are actually more active or more likely to be metabolically active. So with Campbell-Gurley's MitoGFP in the control cells, you can see that mitochondria form this very thin filament around the edge of the vacuoles in subapical cells. In apical cells, of course, they're very active because the cells are growing. And then in the mother cell, they're starting to be active again because growth is taking off again in that cell. But if we look in the RSR1 mutant, you can see that the mitochondria retain this very complex morphology, indicating that they're probably active in all of the cells in hyphae that are subapical. And again, if we look at fluorescence intensity in these two boxes, we can see that there's a lot more intensity in the RSR1 mutant compared to wild type. So where is this taking us? Well, one of the striking features of RSR1 mutant is that it's a multinucleate organism. And if we watch this movie here, we can see one of Tina's lovely movies showing us how the nuclei migrate around in the cell, and they travel backwards and forwards through the open septum, remembering that the septum is delayed in its closure. So Tina followed these individual nuclei and tracked them and numbered them 1, 2, and 3. And we saw a whole bunch of things that we never see in wild type hyphae. First of all, we saw obviously multinucleate cells, but we saw asynchronous nuclear division. We saw a nucleus going into the daughter cell and dividing in there instead of in the mother cell. And then we saw these daughter nuclei didn't migrate at all into the daughter cell. And then here we have a nucleus migrating into the daughter cell without division, but we've got synchronous division of the remaining 2 nuclei. And then a last example here is that this daughter nucleus divides in the mother cell, in the daughter cell, but it then translocates back into the mother cell and then back into the daughter cell. And this, we think, is probably because the septum is open for so long. So how does this affect the overall morphology of the hyphae? Well, what we see is that apical dominance is lost in the RSR1 mutant. So as I was explaining, normally we would see after about 4 or 5 hours growth, you've got the primary growth axis here, but we see an additional branch forming, and that's near the mother cell. Whereas in the RSR1 mutant, you can see that we've got 5 additional growth axes formed in this mutant. So here is a number of secondary growth axes in this mutant. Now, does this mean that actually this is a much better way for the cells to grow, and they would be much more successful if they had evolved to grow like this? But actually, that is not the case, because what happens with the RSR1 germ tubes is that they have a very strong tendency to burst. So if you watch that movie, you can see the cells bursting, and about 60% of the cells will burst. And you can see they try to form hyphae because they've got a Spitzenkörper, but most of them don't make it, unlike yeast, which don't burst at all. And you can ameliorate this by adding sorbitol, which suggests that it's a cell wall deficiency, and that's what is going wrong with these RSR1 mutant cells. So now I'm going to put together for you the mechanism by which we think our RSR1 mutant loses its apical dominance. So here we have the RSR1 mutant, and we know that it works through CDC42 and CLAR4. And what we see is fragmented vacuoles because they can't fuse, because CDC42 activity is not localized properly. And we have retention of cytoplasm, and we have active mitochondria. So then that leads to the cell size checkpoint being inappropriately activated in subapical cells. And when that happens, we get multinucleate cells, and also we get, because we don't have RSR1 function through CDC42 and CLAR4 properly here, we get the septin rings mislocalizing and not assembling correctly. So then the downstream effect of that is that we get DBF2 released from the spindle pole bodies, but it can't associate with the septin rings correctly and makes these clouds, and this delays actomyosin ring contraction, which means that the nuclei can migrate backwards and forwards, and when the septum closes, they get trapped wherever they are. And then that in turn leads to hyperbranching and loss of apical dominance. So then we took the RSR1 null and we transformed it with all the mutants that we had made, all the mutant forms of RSR1 we'd made, and we looked to see whether they could rescue these phenotypes. So here are all our phenotypes down the side here, and then here are our mutants we tried to express. So yellow shows that the phenotypes were all the same as the RSR1 mutant. So the hyperactive form, and if you deleted BUD2, which is the gap, you saw the RSR1 phenotype. But if you deleted BUD5, the activator, surprisingly, we only saw the phenotype in yeast cells. So this is saying to us that either BUD5 is not needed in hyphae, or that the GTP cycling isn't needed in hyphae, but we know that it has to be deactivated. So then we look to the GDP locked version of RSR1. And what we found is that it's only a subset of these phenotypes that actually need RSR1 to be cycling. And we have two phenotypes down here, multinucleate cells and apical dominance, which actually can be rescued by expression of GDP-locked RSR1. When we expressed the palmitylation mutant, we found that it could rescue pretty much all of the phenotypes, showing that our RSR1, if it can function at all at membranes, however transiently, and from endomembranes, then actually the cell isn't in too much trouble. So it may be that the RSR1 can signal very effectively from these membranes. With the farnesylated mutant, if you remember, we think that that's cytosolic because it can't get into the secretory pathway. Again, we find that like the null, it has a lot of phenotypes, but like the GDP locked version, it can rescue the multinucleate phenotype. So the GDP locked version and the version which never gets activated seem to share this phenotype. Interestingly, we then looked in the literature, and where this was missing, we did our own experiments and looked at CLAR4, and we found that CLAR4 shares pretty much all the phenotypes as the RSR1 null. So this confirms our hypothesis that CLAR4 is acting downstream of RSR1 in all of its activity that we've managed to characterize. So then I'm going to map onto you what the null mutant does again and where the rescue points are. So none of the phenotypes are rescued by hyperactive RSR1, but if you express the farnesylated version, you can rescue fragmented vacuoles and the septin ring mutations. And then as a result of that, you get normal regulation of nuclear division and cell division. However, when you express either the GDP-bound version or the GDP version that is cytoplasmic, then you can, what you do is, even though you've got fragmented vacuoles and mislocalized septins, you can stop the inappropriate activation of START and cells become mononucleate, even with these other quite severe phenotypes. So I'm just going to briefly go through the lessons that this study has taught us. So RSR1 is more than just a polarity protein. It regulates a whole bunch of spatial organizational processes within the cell, as it does in mammalian cells. Secondly, it seems to work entirely through CDC42 and CLAR4 functions. Thirdly, the Bud5-GEF is required only in yeast. So we are now looking for other GEFs that might activate RSR1 in other cell functions. Next, RSR1 GDP seems to be required as of itself. So it's probably inaccurate to call it an inactive version of the protein. It does have a function, and at some point in the cell cycle, RSR1 has to be GDP bound to suppress inappropriate activation of nuclear division. And lastly, contrary to the dogma that RAS family proteins have to be membrane bound, we found that RSR1 can actually signal from the cytoplasm. And I was so taken aback by this that I contacted several people to see if they had seen this. And Deb Hogan has also seen this for RAS in albicans, and Mark Phillips has seen this for Drosophila. So this is a new phenomenon that I think means that we have to rethink how signaling occurs. Even though we think it's at the membrane, it can actually be effectively done from the cytoplasm. So then, to thank my group, Tina did the vast majority of the work on this paper, sponsored by the Society for Experimental Biology and now the Wellcome Trust. And Emma did some of the cell bursting experiments, and she's now a student with me at Exeter. And this is the rest of my team who are working with me, looking at hyphae and how they cause the damage that they do. I'd like to say huge thanks to all these people who've given me strains and advice on the manuscript and some of the ideas within it. And of course, past group members who have also contributed hugely to this work. All my colleagues here at the MRC Centre in Exeter, and all my funders. So thank you very much for your attention.
Great talk, Al.
Right, thank you, Alex, for this talk, and we already got a lot of positive comments for both talks, and we are spookily in time. I got a schedule on where we should be, and it's just 1 minute too late, so that's really cool. And now we start with the discussion, and there is already a lot of questions, and Jay, I suggest that Sarah and you start with the first block of discussion, and then if you need a break, I'll take over together with Alex.
Perfect. So the first question, Sarah, is a technical question regarding just basic medical mycology. So given the diversity of the bacteria and other components of the oral flora, how do you selectively enumerate Candida in the oropharynx?
Oh, I see. Yeah, okay. So in the mouse model, we infect mice for an hour and a half under anesthesia with a sublingual exposure. And that's enough for a susceptible animal to make them susceptible. When we sacrifice the mice at whatever time point, if we're measuring the fungal burden and we remove the tongue, we usually take half for RNA or other types of analysis. And then the other half, we basically dissociate completely and then do serial plating with antibiotics to suppress the rest of the flora. Great,
thank you. And then Johannes Westman asked about, could other pore-forming toxins like streptolysin, listerialysin elicit a similar response? And along those lines, do you have any evidence that there's necroptosis and release of IL-1 alpha, since IL-1 may be required?
Right, sure. So the first half of that question, the same pattern applies in PSM alpha. So the Staphylococcus pore-forming peptide PSM alpha, when it infects the skin, induces IL-1, IL-36, which activate mostly gamma deltas, but there is also, if you look closely, a small population of what might be, I mean, there's certainly TCR alpha beta expressing cells, and the nature of their T-cell receptor is not clear. And those were a pair of beautiful papers in Cell Host Microbe just around the same time as our story. So I think it's this principle of inducing cell damage and then triggering a kind of, I don't want to say non-specific, I mean, an early emergency pro-inflammatory response is a common principle. And I will actually say, related to this, Nikki Mitsopoulos at NIH had a story in the periodontal innate, again, TCR alpha beta innate T cells in the periodontium, and they're activated by damage from mastication, so chewing and mechanical damage, but it's the same principle. And so I definitely think this is a common way that you get bursts of inflammation in response to a physical or damage-inducing event.
Okay. And then the second question from Johannes is just the issue is any evidence that IL-17 could play a role in macrophage function, either directly or potentially regulating chemokines that would be important for macrophage recruitment?
Yeah, so I think it's probably more indirect than direct. We do see induction of MCP1, so CCL2. In the mouse model, we don't see a huge macrophage influx or activation, but I don't want to say that, I mean, they're certainly important in fungal clearance. It is possible that 17 acts directly on macrophages as well. It's one of the few hematopoietic cells where I've seen really convincing evidence that they can respond to this cytokine, but we have not explored that at all.
And then one last question, and we can move to Alex's talk, is IL-22 involved just in normal oral tissue structure? I mean, to my knowledge, the IL-22 knockout has a normal gastrointestinal epithelial tract. And
yeah, as far as we can tell, the mouth is normal too.
Right.
At baseline, yeah.
Great. Oliver, let's...
Sure. So let's switch gears. And Alex, I'll start with the first question. And this is, you showed that R1 has this farnesyl rest added. Does this something have to do with the farnesyl quorum sensing system? Is there an affiliation?
I don't think there is, because my understanding is farnesyl inhibits hyperformation. And it's a very different molecule to the long chain fatty acid that is attached. And also, I think that the process of sending RSR1 round the cell farnesylation in the cytoplasm is going to happen to so many proteins that it's just part of the circulatory pathway. So I don't think it's acting as a signaling molecule, but it's certainly something that we could see whether we could rescue something, for example, like that, but not by just mutating the cysteine target site, I don't think.
Okay, then there is a lot of interest in the role of the cytoskeleton, specifically the actin cytoskeleton. Is this the part of the cell that mediates traffic of the nuclei, and does RSR1 directly affect actin?
So RSR1 will sets up cell polarity via CDC42, and CDC42 does all the actin cytoskeleton signaling through BNI1. Now, RSR1 is the activator for CDC42, but when we knock out RSR1, we still see streaming of other molecules backwards and forwards on the actin cytoskeleton. So in Cerevisiae, there is an alternative activator of CDC42 called BUD3. So it may be that RSR1 is not exclusively required for CDC42 activation, but once CDC42 is activated, it sets up the cytoskeleton. However, having said that, I know that people are interested in also whether that's involved in nuclear migration. Nuclei migrate via microtubules in albicans, not by actin. So we see no difference in the actin cytoskeleton, but in the microtubules, we still see them form, but we believe that in the RSR1 mutant, the ends of the astral microtubules are not placed correctly on the plasma membrane, so that the nucleus is being pulled backwards and forwards instead of just unidirectionally as it would normally be. So that's another thing that we are interested in looking at.
Okay, then the thing you showed with the bursting of the cells, can you elaborate a bit on this? Why do the cells burst? And of course, a question that is close to my heart, does this indicate that there is a potentially druggable target, something you could use for therapy?
Well, my model, bringing together what what's in the literature and what Cheryl Gale's papers subsequent to our original paper have shown, is that in the RSR1 mutant, CDC42 is present in the hyphal tip to the same extent as wild type, but it's not focused. So instead of having a very tightly defined growth site where everything's brought together and work as it should, the whole of the CDC42 activity is spread out. And so I would hypothesize that the cell wall generating machinery, so that's the chitin synthases, the beta glucan synthase, they are not functioning in synergy. So you're getting a weakening of the cell wall. And we did the sorbitol experiment to show that it was a cell wall effect rather than I don't really know what else it could have been, but it could have been a cytoskeleton effect, something like that. But we do think it's the cell wall because it's rescued by sorbitol.
And could it be drugged?
Well, RSR1 is the homologue of RAP1 in human cells. And unfortunately, all the systems that I work on, while it's delightful to be able to elucidate what might be happening in mammalian cells and be able to add new perspectives to that, like with our finding that this GTPase can act from the cytoplasm, which goes against all the current understanding about how these things function. So it's great to be able to contribute to our general knowledge about how eukaryotic cells work. Unfortunately, all the systems I work with are conserved. So we would probably, I think the answer is yes, it would be great, because again, we're back to the fact that fungus has a cell wall and human cells don't. So if we can find something in there that that targets one of the mechanisms within RSR1, which doesn't exist in mammalian cells, then the answer would be yes. But it's not immediately obvious how we could do that.
Right. Then I have a question here, wondering whether a regulation of RSR1 function is also done on the transcriptional level. Is there expressional regulation?
I believe that Ryan Ames has looked at that. Because we work with hyphae and hyphal growth almost predominantly, we haven't looked at the morphogenesis program, because for us, if it doesn't make hyphae, we're not really interested in it. So the answer to that, I'm afraid, is I don't know what happens to the transcription of it.
Okay. And then I have a very specific question here, and this is again with regard to the bursting of the terminal compartment. And the question is whether this is only seen when the germ tube is first emerging, or is this somehow related to length of the hyphae? Yes,
that's an interesting point. I didn't mention this, but what we see is that RSR1 the RSR1 mutants germinate much faster than wild type cells. So that again signals to me that the whole thing is disorganized. Sorry, I can't remember the question now.
Yeah, if it's related to the length.
Right. Okay. So I think that it's this initial assembly of the growth machinery that is at fault. Once the cell seems to get past a certain time point, which is well before the first septum starts to appear, if it gets past that point, then off it goes, and yes, it grows. But it seems to be that there is a point at which possibly when the septum starts to form, that's when the bursting happens.
Okay, so thank you very much. And I think we'll switch back, and Jay, you can ask a few questions to Sarah again.
Sure, thanks, Al. So Sarah, we have one question on the S65L mouse model. Any evidence for sex differences or aging in the mice or the humans? Oh, you're muted, sir.
Sorry, just realized that. So in humans, I mean, it was a very small kindred. I don't think there was enough to make a comment, but it was both sexes, and in mice, we see absolutely no difference. The data I showed you was mixed.
Okay. And then what about, is IL-22, is it directly regulating IL-17 receptor expression, or is it just inducing a cell population that then a second signal is regulating IL-17 receptor expression?
Yeah, the 17 receptor itself on a cellular level doesn't seem to change much, right? So we've seen, so 22 doesn't affect it, 17 doesn't affect it itself, other than to transiently lose it when the receptor is internalized. So I think it was really, for us, when we first saw this on the arrays, I thought, how is 22 regulating the 17 receptor? And we went and looked directly. And I think it's just by replenishing the whole cells, it's replenishing that receptor along with everything else those cells are expressing. That's certainly our model at this point.
And is there any evidence of biased agonist signaling with IL-17 receptors? So whether the receptor is engaged by an AA homodimer, an FF homodimer, or AF homodimer, does it cause different conformational changes in the receptor that would affect signaling?
Yeah, a fabulous question. Something we would love to try and get at. It was part of the motivation for making the mice, right? Which was not to prove that it caused Candida necessarily, but to try and get some insight into how these different mutants might work. There's a news story suggesting that an IL-17RC homodimer binds to 17F and maybe can, well, wasn't clear if it could signal, but it could bind to it, and that the configuration of binding was quite different. The way it sat in the membrane was different. And I think, all of this stuff is quite poorly understood, not just for 17 receptor cytokines, but most others as well. I guess kind of implicit to that question is, does 17A and F, even though they use the same receptor, can they activate different signaling pathways? Most of the evidence is that they don't, and that basically A can do things more strongly than F. But there are hints out there that there are things that F can do that A can't. And I think it's very worth exploring in more detail. So if there's any students out there who are excited about signaling and cytokines, let me know.
And then lastly, we have a question from Alistair Brown about what turns this whole process off. So is it MBD3 killing Candida and then clearance of Candidalysin from that perspective, or is there some also host component, resolvins or some other component that helps regulate the resolution phase?
Yeah, absolutely. I'll just hit my very favorite topic. So I'll try to restrain my answer. I mean, I think the first and foremost, it's that as the fungus gets cleared, there's no longer the stimulus to trigger the whole pathway. And in the adaptive setting, that would probably be more from the beta glucans, and in the innate session is more from Candidalysin or some combination. All the from a cell intrinsic signaling point of view, from the cytokine signaling point of view, which is the way I tend to think about things, cytokines, acute acting inflammatory cytokines turn responses up quickly, but they also have a lot of mechanisms in place to shut them down just as quickly, which is a big part of what we do from a signaling perspective. And so for IL-22, that would be like the SOX proteins that inhibit the JAK-STAT pathway. In the IL-17 receptor, there's a lot of regulation is at the RNA level. So the RNAs that get it turned on, like IL-6 and CCL20 and so forth, are very intrinsically unstable. And a lot of the genes that we see in these arrays are negative regulators of RNA. And I could go on for hours, but I won't.
Thanks, Oliver.
Okay, so thank you very much. And again, we are perfectly in time. I'll take the chance to read the comment by Liz Ballou. Thanks to both Alex and Sarah for great talks and fascinating discussion. I think that's what we all feel. Thank you very much. I also want to take the chance to thank the MRC team for a wonderful organization. This has been really professional and no mistakes, no problems, nothing whatsoever. And then the final word is, please remember the next Myco-Talks on the 19th of November. Again, very fabulous speakers, so be sure to mark this in your calendar. Thank you very much for joining and have a nice day.
Thanks, Alex.
Thanks, Oliver and Jay.